Okay. Good evening, students. So, welcome you to the fourth tutorial for this course, Introduction to Cell Biology. Hope that you have gone through the contents of the fourth week of the course. So, as always, we will try to look once more into the summary of what has been taught in this fourth week of this course. So in this fourth week, actually, the focus has shifted from molecular biology to more of cell biology. So primarily in this week, the focus was to know more about the different types of cells, prokaryotic cells, eukaryotic cells, and their nature of organization. The nature of organization of the cellular biochemistry within the prokaryotic cell and the eukaryotic cell. Now, before that, uh, you have been given, very justifiably so, an introduction or an overview of the microscopic techniques that has made possible the investigation of cells. That has made possible, I mean, that has made possible cell biology at the first place. We have no other, I mean, microscopy turns out to be one of the major techniques because seeing is believing. So for getting the knowledge about cells that we have today and even using microscopic techniques to know more about different cellular aspects as we go ahead. So among the different microscopic techniques, the most common microscopy is light microscopy. So on the left hand side, you can see the simple light microscope that was first developed by Leeuwenhoek, which allowed him to see very small objects which were otherwise not visible. And however, this light microscope that Leeuwenhoek built for the first time is more of like a magnifying glass. So it gives you magnification. It shows you the small organisms which are not visible to the normal eye in a more magnified manner so that they become now visible. But it consists of a single lens, single magnifying glass like setup. Whereas on the other hand, the more complex model is the compound light microscope. Um, so in the compound light microscope, as many of you already know, that this has a complex in comparison to the light microscope, the compound microscope has a much more complex set of arrangement of several lenses. For example, there is this condenser lens. So there is obviously in case of light microscope, the source of illumination is visible light, and there is a source of light, and then there is this condenser lens which focuses the light on the sample that can be mounted on the stage, and from that the light comes to the objective lens which can have different magnifications. You can have 10x, 40x, 60x, 63x objective lenses, and from this eyepiece lens or the ocular lens, after which you can place your eye on top of this eyepiece lens and eventually you get to see the magnified image. So this compound microscope has a much more complex set of several different lenses and actually provides much higher magnification capabilities as compared to the light microscope, which is more of like a magnifying glass. Now, there are two things that should be clear in microscopy. These two things are magnification and resolution. So the thing is that just magnification is not enough for imaging and for seeing very small objects, because magnification literally means that if that, for example, if a very small object and your optical setup is allowing you to see an image is a much larger version of the actual object, right? So that is what you call magnification. But literally the magnification, the formula can be kind of described in the form of an equation that magnification is equal to size of image divided by the size of the real object. However, just magnification sometimes may not help, because may not help if you do not have the resolution, because resolution is something which means, for example, you have two very closely spaced objects. Let's assume that these are the two dots which are your objects, so which are located very close to one another. Now, resolution means that whether you are capable to distinguish and observe these two objects as two individual objects or not. If you are capable of seeing these two objects as two different entities when you are magnifying, then you are capable of resolving. You have that resolution. However, if there is one resolution limit, so resolution limit means that, for example, here you have this distance, these two dots are placed at a distance x. So when they are placed at this distance x, you are capable of, when you are magnifying, you are capable of also distinguishing between these two dots as two different objects. But beyond a certain point, for example, when the distance is x by 4, that is, they are even located, they are located closer to one another, even close. Then you are not capable, for example, you are not able to distinguish them as two different dots. Instead, you are seeing them as a single dot. So this is where resolution becomes important. So here you will see it as a single dot. However, it is not actually a single dot. It is actually two different dots which are located very, very close to one another. So close that it is beyond your capability of resolution. That's why you will be seeing it as a single dot. So not only magnification, but along with magnification, the resolution is also important, so that you are capable of distinguishing two different objects which are located very close to one another as two individual different objects. Now, there are several factors on which this resolution depends. So basically, resolution can be described in other words as the minimum distance. Okay, the minimum distance uh, uh, between two objects at which you are capable of uh, kind of differentiating or observing or distinguishing those two objects as two objects. You are not seeing them as single object. So the distance between two objects at which you are capable of seeing them as two objects, distinguishing them as two objects. Their individuality is very clear. So that is the resolution. So beyond a certain distance, you will not be capable of distinguishing them or seeing them as different objects. You will be seeing them as a single object. So that is beyond your resolution capabilities. So that distance, this is what is being represented here as delta x. So that depends on several factors. For example, the wavelength of light, it is directly proportional to the wavelength of light and inversely proportional to several different parameters. For example, the refractive index of the medium, the refractive index of the optical medium between the objective lens and the sample. Okay. So it can be air, it can be water, it can be oil. The oil immersion objectives. So in that case, the oil gives you a different refractive index. So these media have different refractive index and gives you different resolution, right? And it also depends on this parameter, which is being said here as sine alpha. So what it really means is that when your objective, so this is your objective lens, this is your objective lens, this is your sample. So the light that is being collected by the objective lens is in this form of a cone, right? So if you draw a perpendicular from this sample, then this angle is your alpha. So this resolution also is inversely proportional to sine of this alpha. And actually, this n sine alpha is together given one optical, is represented as one optical parameter or optical property of the objective lens, which is called the numerical aperture, right? This is together referred to as numerical aperture. So essentially, the resolution capability, the resolution that is being achieved by a set of objective lenses by a set of objective lenses is directly proportional to the wavelength that is being used and inversely proportional to the numerical aperture. That is this alpha angle value. So for example, this alpha is also dependent on this working distance between the objective lens and the sample. For example, if you have 10x, then if this much is the distance, and from 40x, what happens is the objective lens operates at much closer working distance. So in that case, what you will see is that the cone is a bit wider. That is, the alpha value is bigger. So sine alpha value is also bigger. So numerical aperture is bigger. That is, resolution becomes better. Lesser is the distance, the more will be the resolution. The lesser is the distance means that even if the two objects are far more closer, still you are able to resolve it. So yeah, so for different objective lenses, for example, 10x, 40x, 100x, so greater is the magnification. Also, what happens is the objective has to be operating from much closer distance. It has to be placed much closer to the sample. And so what happens is that this cone also becomes much wider. The numerical aperture becomes much wider because of the increased sin alpha. And the refractive index, what you do is, for example, 63x objectives and this 100x objective, you use oil, which has higher refractive index. That also increases the numerical aperture, and therefore they give you better resolution. They decrease this distance, which means that even if the two objects are located far more closer, still you are able to resolve them. However, all of this is possible only till a certain extent. Like that's what is referred to as the Abbe's diffraction limit. That if you're using light as the illumination source, then light has a wave nature and it faces diffraction. So there is a limit beyond which you cannot. Um, there is certain distance which is actually 0.2 nanometer, beyond which. So for example, if two objects are placed at 0.2 nanometer or even closer, less than 0.2 nanometer, 0.1 nanometer or 0.05 nanometer, then you will not be able to distinguish those two objects as two different objects. You will be always seeing them as a single dot. You will not be seeing them as two dots. That's what is referred to as Abbe's diffraction limit, right? So as it is mentioned here, there's a diffraction barrier. So only if these two dots, if you are operating at a max capabilities, for example, using 100x objective, you're using oil as a refractive index, you're operating at your maximum numerical aperture and at a certain wavelength, then even after operating at a maximum capability, you can, you will get a theoretical like distance which is less than 0.2 micron, but since you're using light as the optical illumination source, if two objects are placed at a distance which is less, which is 0.2 micron or lower, then you will not be able to distinguish those two objects as two different objects. You will be seeing those two different objects merged to form a single object, right? That is, it will be beyond your resolution diffraction capability. That is this because of the diffraction limited nature of light. So for that, what you will need is you will need to change the illumination source. You will need electrons as your illumination source, right? That's why electron microscopy gives you much higher resolution as compared to light microscopy. Okay, this we have already mentioned. So this slide gives you an idea of the different ranges of magnification and resolution powers of different techniques. Like you can see what is the magnification and resolution capabilities of just the naked eye, light microscopes, and then electron microscopes, right? So as the wavelength goes lower and lower, so the resolution actually goes higher and higher, because you are capable of resolving two objects which are even placed at lower distances or even more closer. Right. Now in optical microscopes, like optical light microscopy, there are some other additions or some other optics that has been invoked, like in addition to the compound microscope, the compound light microscope that we saw, where we had one condenser lens, then the sample, and then the objective lenses, and then the ocular lenses. So some more optical components and optical techniques and technologies have been used to devise some more optical microscopic tools that provide you much better contrast and allows you to differentiate different subcellular compartments having different optical densities or different optical properties, refractive indices. For example, this phase contrast and differential interference contrast microscopes. Right, bright field is the simple. Is the second compound microscope, compound optical light microscope that we saw. Phase contrast and DIC. What is they take advantage of these different refractive indices of different subcellular compartments. For example, the nucleus is much more dense as compared to the cytosol. So light that passes through the cytosol versus light that passes through the nucleus have different phases, right? Light that comes out of these two different compartments of the cells are in two different phases. That creates a phase contrast between these two subcellular compartments. And in this phase contrast microscopy, because of certain special optical arrangements, which further enhance, which capture basically this phase gap between the light that is coming out of the cytosol versus the light that is coming out of the nucleus, to create a nice contrast between the nucleus and the cytosol to form a much better contrastful image. And same for differential interference contrast microscopy and various other different optical microscope techniques. Now, in cell biology, one type of till now we had been talking about optical light microscopy, where we were using visible light, and however, also even not using monochromatic light. We were using light having wavelengths of so many different wavelengths. Polychromatic light, basically. We're not using monochromatic light. But there is one type of microscopy which has turned out very important for elucidating cell biology, for the knowledge of cell biology that we have currently, and is widely used in elucidating more, in solving more mysteries of the cells, which is fluorescence microscopy, right? So in fluorescence microscopy, basically fluorescence microscopy, as the name already suggests, is microscopy that takes advantage of fluorescence, right? Then fluorescence, what happens is there are certain molecules which are called the fluorophores, which get excited. So basically, these molecules have electrons. So when you shine a light of a particular wavelength, okay, of a particular wavelength, that is light that can provide the electrons a certain amount of energy, then these electrons move from their ground state. The electrons move from their ground state to the excited state, excited electronic state, ground electronic state to the excited electronic state. Now, after that, what happens is when these electrons first undergo vibrational relaxation. So in each of these electronic states, basically there are different vibrational substates. So these electrons actually undergo vibrational relaxation in this singlet, in this S2 or S1, and finally these electrons again come back to the ground electronic state, and the energy and release energy, which is equivalent to the energy gap between this excited energy state and the ground energy state, in the form of photons or in the form of light. So fluorescence is light which actually has an increased or longer wavelength than the excitation light. In fluorescence, what happens is you have a specific excitation for a particular fluorophore. You have a particular excitation wavelength where you have the maximum excitation or maximum absorbance, and also you see light emission or emission of light having the maximum intensity at a particular wavelength. So there is maximum intensity of emitted light at a particular wavelength. So there is this very very controlled and characterized nature of fluorescence that you can use a particular wavelength to excite a particular fluorophore maximally or optimally at its lambda max, and you also get a lambda max of emission. That is that particular wavelength of light where you get the maximum fluorescence intensity, right? Why fluorescence microscopy has revolutionized cell biology is because of this fact that fluorescence microscopy allows us to view, to see or observe specific compartments of the cells, or even to be more precise, just a single molecule or molecules at a molecule by molecule level, or um, at single molecule level specifically. For example, if you want to see the cellular localization or the subcellular localization of one particular protein, so what you can do, you can take two approaches. For example, the one approach will be if you have one antibody that specifically recognizes that particular protein, right? And on top of that antibody, you have attached a fluorophore. A fluorophore that is, for example, a green fluorophore. That is, it will be excited by a particular wavelength of light, and it will emit a particular wavelength of light, right? So then you can use these antibodies to basically label only your protein of interest, right? Only of protein of interest within the cells. So basically your protein of interest within the cells will be labeled by these antibodies, which are labeled by the fluorophore. Now you do fluorescence microscopy, you only see your protein of interest subcellular localization, right? Another way to attach your fluorophore, which can be fluorescent proteins like GFP or M-cherry, etc., to your protein of interest, is to take advantage of the molecular biology tools where you can clone this GFP or RFP or M-cherry, etc. Just to the N terminal part or the C terminal part, you can add this particular protein amino acid this nucleotide sequence which encodes for the amino acid sequence of GFP or RFP or M-cherry to the N terminal part or the C terminal part, that is upstream or downstream of your protein of interest. So to create the fusion proteins, right? That is your protein along with the fluorescent protein, whichever it is, GFP, RFP, M3, etc. So thus when this protein will be expressed in the cells, it will be expressed along with this fluorescent protein tag. So when you will be doing this fluorescence microscopy, you will be able to see the subcellular localization of a protein of interest. As well
Sir,
as, so you can do multiple, Shom.
sir, it is acting like the marker, gene marker.
Yes, yes. Marker means, by marker, what you are truly meaning, can you say?
So marker means one gene is highlighted, one newly inserted gene is
Yes,
given, and that
yes.
gene
One
is
particular,
inserted or not inserted. To understand that, marker is given.
no, not like that. So not like that. Here, what I'm trying to say is, for example, if you are adding a fluorescent protein to only a particular protein of interest, right? So you will see the fluorescence of a fluorescence. If you are using one particular wavelength for excitation, you are collecting the fluorescence at only a particular wavelength. So that can come from only a particular fluorophore, and only that fluorophore is attached only to your protein of interest. So in fluorescence microscopy, it will allow you to see or visualize just your protein of interest very specifically within these cells where so many different molecules are present. That's what I was trying to say.
Okay, sir, understand.
Yeah, so basically I'm trying to say that in fluorescence microscopy, if you are adding fluorophores can be organic fluorophores like Alexa dyes. So Alexa dyes can be attached to antibodies, and now with this Alexa tagged antibodies can recognize a particular protein of interest, And you can use fluorescence microscopy to just visualize the single protein of interest that you are interested in. Or you can add these fluorescent proteins to your protein of interest, and now fluorescence microscopy can be used to see the subcellular localization as well as protein-protein interactions, right, within the cell in a very specific manner. So there are so many molecules within the cells, but you can use fluorescence microscopy along with these, uh, you know, antibody-mediated tagging of fluorophores or addition of fluorescent proteins specifically to particular proteins of interest, which are encoded by obviously the genes of interest, to see them within the cells. Okay, to see their subcellular localization as well as to see their interaction. For example, there are two proteins, protein A and protein B. So you have tagged protein A with green and you have tagged protein B with red. So you can individually take the image in green and red. Now you are seeing yellow. So what does yellow means? Yellow means that red and green are co-localizing. Since they are together in one particular compartment, might be interacting, might not be interacting. You have to do other experiments, but at least you can say that, okay, they are in one particular compartment. So there is a chance of them interacting and maybe driving one particular cellular function. So that is what fluorescence microscopy allows you, and therefore has revolutionized cell biology, because you can do compartment specific visualization of individual protein molecules of interest. Clear? So,
Yes, sir. Clear.
okay. So, moving on. So basically, um, I mean, this is just to show you this is just a schematic of the fluorescence microscopy, or this is called the wide field or epifluorescence microscopy. So just to draw a comparison between the, um, I mean, this compound optical light microscope without, which was non-fluorescent, I mean, not the fluorescence microscope, and this fluorescence microscope. So the overall principle is similar, just that in case of this fluorescence microscope, as opposed to the compound optical microscope that you saw, here there will be use of a single, a monochromatic light of a certain wavelength, which is that particular wavelength which will be capable of maximally exciting the fluorophore, right? Which is with the fluorophore that is present in the sample. And so after excitation, what will happen is that the emission light will be collected, but the emission light will have much longer wavelength. So there is a dichroic mirror which allows this lower wavelength excitation light to come in. It doesn't reflect that, but it will reflect the longer wavelength emission light when the emission light comes in, and it will reflect it towards the direction of the detector, right? Which will collect the emission light and which will eventually give you the image. So basically there is excitation with monochromatic light of a certain wavelength, which passes through the dichroic mirror, is focused by the objective lens on a sample, which is labeled with the fluorophore. The sample has the fluorophore. Now, when the fluorophore is excited, it will eventually emit the fluorescence light, which will have longer wavelength than the excitation light. Now, this longer wavelength will be again collected. Longer fluorescence light, longer wavelength fluorescence light will be collected by the objective lens. It will be reflected by the dichroic mirror towards the first a set of lenses, which will focus it to the um, this detector, which will eventually give you the image, the fluorescence image. Okay. Now, what happens is I will like to introduce one more concept here. So, in this setup, like if you are just exciting your sample, okay, you are exciting your sample with the light, monochromatic light of a certain wavelength, then your sample is getting excited. Sample which is basically labeled with the fluorophore is getting excited and is giving you the light. The emission light, or basically the fluorescence emission light, of longer wavelength is being collected and focused and basically directed to the detector, which is giving you the image. But consider this fact that your sample, the biological samples typically are quite thick. There are several biological samples which are quite thick. That means, and in many cases, it doesn't consist of a single layer. It that has some. So there are three coordinates, right? There are x coordinate, there are y coordinates, and there are z coordinates, which is basically height. So many of the biological samples are there which have several different layers of organization, right? Which are quite thick. That is, they have different information at different z planes, right? Now, in this case, when you are exciting the sample as well as collecting the emission light, you are collecting the like all the different planes are getting excited. Fluorophores, which are present in all these different planes, are getting excited, and the emission light is also being collected from all the different planes altogether. So you are not getting the plane specific information. You are basically seeing an averaged information which is obtained from all the different planes altogether. So you are not able to distinguish between the you are not getting the plane specific information, and therefore you are not able to distinguish between the information that is present in the different planes or different z uh, coordinates. This is something which has been addressed by another setup which has just one very simple addition, but very important addition, which is that type of fluorescence microscopy. So here, in which you don't have that setup, you are collecting the information from all the different planes. That is called the wide field or epifluorescence microscope. So in this, in the advanced setup, in one more advanced setup, what you have is you have one pinhole, right? So pinhole can be literally is the pinhole of a camera. It's like a plate with just a small hole in its center. It can be visualized that way. So that pinhole is placed later um, uh, before the detector, after this dichroic and all these lenses. The pinhole, what the pinhole allows is it allows the light that is coming from just a certain plane, a certain z plane, to focus eventually on the detector. So that pinhole allows you to collect plane by plane information, plane by plane information of a certain plane, and you can change which plane's information you are collecting. So it allows you to scan the entire sample literally plane by plane. So it gives you the capability of optical sectioning and 3D volume reconstitution. So that's where we will also be coming. We are heading next up basically. Is this? This is what I was trying to tell you, that initially we saw the setup of epifluorescence microscope, but in epifluorescence microscope or wide field fluorescence microscope, what happens is that your biological samples which have this depth of focus, right, which have several different layers of organization, right, different information at the different z planes, there epifluorescence microscope or this wide field microscope, wide field fluorescence microscope I'm trying to mean, that is not able to capture this plane specific information individually. It gives you an averaged information where all this plane specific information are averaged out. But if you place this pinhole just before the detector, it allows this fluorescence microscopy. It is called the confocal fluorescence microscopy, which allows you to collect information just from a single plane at a time, and therefore gives you much higher resolution images, as well as that is higher z resolution images, as well as it allows you to uh, do basically this optical sectioning. What does optical sectioning means is optically you can as if section, you are as if taking a knife and cutting this thick sample into very small thin z planes and collecting the information individually one at a time, but you can also stitch them together to create a three-dimensional reconstitution of this entire sample, right? This example will tell this thing much better. I mean, I hope that it will help you. Shubham.
Sir, the cutting of the planes is done by the laser?
No. So it's no, it's not the laser. The thing is that you are exciting the sample using one laser. Okay. Then your sample is emitting, but all the different planes, right? For example, okay, let me tell you that here you will perhaps maybe understand. For example, here you see this red thing. This is basically an epifluorescence image of a pollen grain. Okay, this pollen grain um, uh, actually this pollen grain has two. It's every biological sample is three-dimensional, right? Nothing is. I mean, we see the 2D images, but biological samples are three-dimensional. So in this pollen grain, there is some uh, um, specific proteins which are expressed only by this core part, and then there are this spiny outer part. So the spiny outer part expresses proteins which are labeled in red, whereas the inner, right, the inner core, which is being surrounded by the spiny red part, the inner core is green, and the outer core is red. The outer leaflet basically is red. When you do the wide field fluorescence microscopy, epifluorescence microscopy, you just see the red outside. You don't get to see the inside of the pollen grain. And hence the entire pollen grain is appearing to be red without any Z resolution. Confocal fluorescence microscopy, what you are doing is, you are not cutting the sample with a laser. What you are doing is, you are putting a pinhole. Okay. But basically what you are doing is, for example, just before this light enters into the um, detector, it is focused into the detector. What you do is you put a pinhole. Okay, you put literally like this. A pinhole is like a plate which has a small opening, right? A very small opening in the center. How that helps in optical sectioning and in achieving confocality is by this. So for example, this is your detector. Okay, this is your detector. Now, in case of this epifluorescence microscope, your biological sample has so many different layers, right? So many different Z's. Now, light is coming from these different planes and they're focusing in different positions. Okay. Lights from these different planes are coming in and all of them are focusing all together on this detector. So the detector is collecting light that is coming from all of these different planes all together. It cannot distinguish between the planes. It cannot take the plane by plane information, right? But if you put this this pinhole in the confocal fluorescence microscope, what will happen is that for the same sample, right? Again, light from different planes will come in. Okay. For example, this comes and this focuses like this. So it is not allowed to go through the pinhole. Okay. Whereas, for example, this thing, it focuses like this. So that also cannot go into the detector. Just the light which is coming from, for example, this plane, okay, is capable of going through to the detector. So the light which is coming from just a single plane out of all of these different planes is able to now go to the detector and form the image. So confocal fluorescence microscope is allowing you to take the image of only a single Z plane of all out of all of the Z planes that are present in your sample. And that's why is enabling you to do the optical sectioning. That's what we are referring to as optical section. It's not the laser mediated cutting of the sample. It's the pinhole mediated collection of the fluorescence emission light that is being emitted only from a Z plane of a certain very small width of 350 nanometer, 350 micron to 500 micron. Is that clear?
Yes, sir. Understood. Yes, sir.
Yeah. So because of that, now with confocal microscope, you can see that you can literally, for example, this is your entire sample, for example. So with confocal microscope, you can first take a plane which is here. Then you can take the image of a plane which is here. That you can see here, like 1, 2, 3, 4. So 1 is the lowest plane of the sample. Then you go a bit higher, you get 2. Go a bit higher, you get 3. You go a bit higher, you get 4. You go a bit higher, you get 5. Go a bit higher, you get 6. Like that. So like that, you can literally, get these optical sections. You can scan the entire sample. You can get the very nice 2D images of each plane of this sample, and eventually what you can do is you can put together all these planes, right? You can put together all these planes to create or reconstitute the three-dimensional fluorescence image of this particular sample, where you will be very clearly able to see that, okay, that the core is expressing that particular protein, which is expressed with a green fluorescent protein, for example, GFP, and the outer shell is expressing that particular protein which is tagged with a red fluorescent protein, for example. And you also get the intermediate, these Z planes, these very high Z resolution, nice 2D images of the thick biological sample. Right? So that turns out to be very helpful. Along with this, one more image processing technique actually allows us to considerably improve the resolution of the confocal fluorescence microscopic images, which is called mathematical deconvolution, right? So actually what happens is that when you basically shine this light, or even when you basically collect this emission light, the light is not, uh, because of the wave function of light, the light intensity has an interesting distribution, which is called the point spread function. So is I mean, if you see this, it's like this, there's a central very high intensity light, and then there are these concentric circles with gradually dimming up intensities. And there's a point spread function. So basically, in this mathematical deconvolution, it's a mathematical algorithm which tries to identify just this central very high intensity light band, and this algorithm is applied on these confocal microscopic images. When these algorithms are applied, it gives you much crisper and much better resolution images. Where, for example, even in this image and this image, if you see, this is the raw image. This one is the raw image, left one, where these two different strands seem to be a bit, overlapped with one another. However, so that is because each of these has this point spread functions. Like this is the central high intensity band. This is the central high intensity band, but they have these gradually dimming up intensities, which gives you some extent of background fluorescence in between. So deconvolution has basically cleared up the background fluorescence, which was here and here, and gives you a much more crisp and high resolution and clear images, where you can distinguish between two different objects which are located at very close distances. So this mathematical deconvolution image processing algorithms also help you in de-blurring and getting a little bit more resolution images, confocal microscopic images.
Sir,
Okay, so this was Hajom.
sir, these are the images of microtubules. Previous images.
These are the images of microtubule,
Okay, sir.
cytoskeletal elements. I mean, yeah. So basically, these are actin filaments. Um,
Okay, sir.
because microtubules are so now let's move to the biology of cells. Now, cells primarily are of two different types. There are prokaryotic cells and then there are eukaryotic cells, right? So prokaryotic cells are have much simpler um, organization of cellular biochemistry as compared to the eukaryotic cells. The prokaryotic cells are also much smaller than the eukaryotic cells. So for each cell, just like the eukaryotic cell, the prokaryotic cell is also delimited by a lipid bilayer, right? Lipid bilayer membrane, and after to the outer side of the this lipid bilayer membrane is actually called the cell membrane, and then there are additional layers that lie outside of the prokaryotic cell membrane, which are the cell wall and outer membrane that together consist of the cell envelope. Now, prokaryotic cells are these prokaryotes or prokaryotic cells, which are the first cells to form. Um, uh, they primarily consist of bacteria and archaea. Okay, and uh, like in evolution, this is a trend that since these are the first cells to be formed, they are much simpler, and they were uh, from eventually in evolution much more complex forms of the cells were formed, which are the eukaryotic cells. So in if you look at the organization of the bacterial cell, then as I already mentioned that there is this cell membrane, which is the lipid bilayer, and then the cell envelope consists of the cell membrane, the cell wall, which is primarily made up of peptidoglycan, and in case of the gram-negative organisms, there are additional layers like the outer membrane or the capsules, which together form the cell envelope. Now, within this cell membrane, the bacteria primarily has two compartments. One is the cytoplasm and this nucleoid. There are no other membrane-bound organelles or subcellular compartments. There are no subcellular compartments apart from this nucleoid and cytoplasm. The nucleoid is also not membrane-bound. Nucleoid is actually the bacterial chromosomal DNA, which stays in a highly compacted manner because of the nucleic acid or the bacterial chromosomal DNA and protein interactions. Okay, that forms a density of compacted bacterial chromosomal DNA within the bacterial cells, which is called the nucleus. And then the rest is the cytoplasm, which also has the bacterial ribosomes, right, which are the bacterial protein translation machineries. And bacteria also has many of these locomotory kind of structures that are emerging out, which are the flagella and the pili. However, pili also has important roles in adhesion of the bacterial cells to the various environmental surfaces, as well as many of these pili, you may have already heard, that are involved in horizontal gene transfer across bacterial cells. Yeah. So now, basically, if you try to think that what are the sources of energy for the prokaryotes, then some of these prokaryotes, bacteria and archaea, they are photosynthetic, that is, they are autotrophic, so they can harvest sunlight, that they can harvest the energy from sun, harvest sunlight energy, and can convert it to chemical energy. Whereas many of these bacteria and archaea are also heterotrophs, that is they depend on that is they feed on, many of them are parasitic, that is they feed on organic matter that they obtain from various other living organisms, like many of these pathogenic bacteria, and many of them are saprotrophic, that is they basically feed on dead decaying organic matter, and they also play important role, however, in the ecosystem and ecology, because they are these decomposers and play important roles in the recycling of nutrients. Eukaryotic cells are much much complex, much bigger than prokaryotic cells. They have a much more complex organization of the cellular biochemistry, in terms of, because in the eukaryotic cells, we have several membrane-bound organelles, where within each organelle, like within different organelles, we have different sets of proteins and different environmental conditions and different functions that are being executed. Simultaneously, all of these different functions are being executed within the same cell. But within this, because of the presence of these various different subcellular membrane-bound organelles, right? So, and in case of the eukaryotic cells, even this genetic material, that is the chromosomal genetic material of the chromosomal DNA, is present within the nucleus, which is a double membrane-bound organelle. So, whereas in case of bacteria, you had the nucleoid, which is membraneless, and where this density, this nuclear density arises simply because of the interaction of the bacterial chromosomal DNA and the bacterial proteins, which lead to the compaction of the bacterial chromosomal DNA. In the eukaryotic cells, you have the chromosomal DNA located within a membrane enclosed, double membrane enclosed nucleus, right? Prokaryotic cells are, almost all the prokaryotic cells are unicellular, and the prokaryotic cells also reproduce by binary fission, right? Like, so basically their reproduction itself is by binary fission, because a single prokaryotic cell is equal to an organelle. Prokaryotic cellular. So binary fission, the cell division is equivalent to reproduction. Whereas eukaryotic cells, um, eukaryotic cells actually form multicellular organisms. That means a group of eukaryotic cells uh, get differentiated into tissues. A group of eukaryotic cells can communicate with one another and can function like a group, coordinated, cohesive group, which is which are the tissues, right? Then these tissues can also form the organs, and organs can form organ systems, which eventually form these multicellular large organisms, like human beings, dogs, cats, and others. So eukaryotic cells can be multicellular, and thus in those multicellular organisms, also see reproductive cells which actually give us gametes, and then there is fertilization, and that's how the reproduction proceeds. So just cell division is not equivalent to reproduction. Eukaryotic cells obviously include both animal cells and plant cells, right? Now, this is the thing that one thing is that the eukaryotic cell is highly sub-compartmental into so many different membrane-bound compartments. So the eukaryotic cell primarily consists of this plasma membrane, this outer plasma membrane or cell membrane, which is a lipid bilayer that delimits the cell, and the existence of the cell is primarily because of the existence of the this lipid bilayer plasma membrane, which also is very important in maintaining the cellular homeostasis of so many different of the molecules that are present within the cell, the molecules that come in and come out of the cells, and the overall homeostasis of the cell. And in addition to the plasma membrane, within the cell, there are so many different subcellular membrane-bound compartments, like the mitochondria, the endoplasmic reticulum, the nucleus, the Golgi, the lysosomes, the peroxisomes, the centrioles, etc. Where within each of these compartments, you have very different sets of proteins and very different um, environmental conditions as well, very different molecular packaging, molecular crowding, and because of which you are capable of these cells are capable of executing so many different cellular biochemistries, so many different cellular biochemistries that need so diverse and different environmental conditions as well as protein players. But all of these functions can be executed simultaneously because of the presence of this membrane bound compartments, within which you can have this specific enrichment of so many different sets of protein players, as well as so many different chemical environments which are necessary for executing the different biologically important biochemical reactions, right? And also consider this fact that within a multicellular organism like human being or even dogs and cats, there are so many different types of cells that we have. And then, so basically cells have differentiated into different types of cells, and there are also cell to cell differences and cell specific subcellular compartmentalization differences. For example, there are some cells are called the polarized cells, whereas other cells are called non-polarized cells. So polarized cells, for example, the neurons or these epithelial cells, where there is one apical membrane and one basolateral membrane. So on end you have this villi, whereas on other end you have the basolateral end, which faces the blood vessels to which the nutrients are passed on. So the nutrients can pass through the entire body. In neurons, which are also polarized cells, you have the soma, the dendrites, and the axon, which are very different functions, right? And which also need very regulated subcellular compartmentalized activities for carrying out the different functions. So yeah, so there are also cell-to-cell differences in the subcellular compartmentalization and subcellular compartment-specific functions and how they're regulated eventually for the normal physiology of the tissues, organs, and eventually the entire organism, right? So yeah, that is a thing that I mean, we have mentioned it quite a few times that the eukaryotic cells are quite complex and they have so many different uh, subcellular compartments, membrane-bound subcellular compartments. And within these subcellular compartments, they have different sets of executor molecules, different biochemical processes going on. But there is also a cohesiveness or concertedness or coordinatedness between all these different compartments. None of these compartments are like working in isolation. It's not that um, one compartment is not talking with the other compartment, whereas it's actually a very cohesive and concerted process where all of these biochemical reactions and processes going on in different cellular compartments are very highly coordinated by master regulatory circuits, and is very similar to this kind of traffic map of any city for that matter. um, but there are very small units where each of these units do their own functions, and but eventually also there are master regulators which oversee the entire process and ensure that all of these small units are working in a concerted, coordinated manner for ensuring the effective functioning of the cells and eventually the groups of cells, that is the tissues, and eventually the organs and therefore the organisms, right? There is considerable crosstalk and master regulation of for cohesive and concerted activities within different organelles of the cell, for cohesive and concerted communication between all the cells that are present in certain tissue. There is cell-cell communication, and then there are also like organ-to-organ communication, right? Yeah, for maintaining the overall physiology. So now let's delve a bit deeper into the different subcompartments of the eukaryotic cell. So the first subcompartment of the eukaryotic cell is the cell membrane or the plasma membrane, which is the outermost layer of the eukaryotic cell, which consists of the lipid bilayer, right? It consists of the lipid bilayer. This lipid bilayer is primarily made up of the phospholipids, but along with that also has cholesterol, sphingolipids, and various other and obviously several different proteins. It's like the according to the fluid mosaic model, it's like the proteins that are floating in the sea of lipids, right? Lipids primarily phospholipids, but also having cholesterol, sphingolipids, and several other glycolipids, etc. These proteins, this plasma membrane plays very important role in maintaining uh, the cellular homeostasis, like the internal cellular conditions, right? And without the plasma membrane, there is no existence or identity of the cell. So it is only it is a plasma membrane because of which the cell can function as a independent functional unit. Not an independent. I mean, independent. It is independent, but also in communication. I mean, it's in constant communication and this coordinated um, dialect or dialogue with all the cells that form the tissues and eventually organs. But yes, it makes the smallest functional biological unit. And these proteins that are present in the plasma membrane play an important role in ensuring that there is continuous exchange of molecules between the cell and its external environment. Molecules can come in and molecules can go out, but in a very selective, permeable manner, right? Okay. So the next important subcellular compartment is the nucleus.
Sir,
So the nucleus, uh,
sir,
has home.
sir, can you give one example of the organ complication you told? Organ complication, can you give
Organ.
one example?
It's very simple. For example, you take the example of these hormones, right? So in hormones, what happens? For example, if you think of this system, that there are several of these communication systems where you have, for example, gut-brain communication system, you have kidney-brain communication systems, right? So, or you have multi-organ communication systems where this kidney, heart, and brain come together. I mean, where the circulation is controlled accordingly. Or, I mean, for example, you feel this like after you eat a certain amount of food, you feel satisfied, right? So that is because when you after you have eaten a certain level of food, your stomach muscles are stretched to a certain extent. Then there is a signaling between the stomach and the brain, which eventually makes gives you this feeling that okay, I am done. So that is called satiety circuitry. So for even for example, the kidney system that I am trying to mention actually is called RAS, renin angiotensin system, RAAS. You can look into that. So that is also a system where this kidney, brain, and I think also heart communicate with one another because of signaling that is being done via these soluble molecules that are being secreted under a certain situation. Or for example, you take this very well-known example of insulin for that matter, which actually controls your glucose homeostasis, right? By elucidating a very controlled response between the muscles, the fat storage organs, and the liver. So
Okay,
these are
sir.
also part of
Understood.
organ-organ communications.
Yes, sir.
Yeah.
Understood.
So the nucleus is an important subcellular compartment which actually harbors. So the nucleus is actually a double membrane bound organelle. So how it is? It's quite interesting that the membrane nucleus actually is like this. It has uh, it's like this. Okay, wait. Actually, the nuclear membrane is double membrane bound. Okay, and actually the nuclear membrane is continuous with the endoplasmic reticulum membrane. Okay, nucleus membrane continues to form the endoplasmic reticulum membrane. That's why you will always see the endoplasmic reticulum to be located just adjacent to the nucleus, because the nuclear membrane continues to form the um, endoplasmic reticulum membrane on the cytosolic end. And these gaps that I have left are actually not gaps. This has multi-protein complexes okay, which are present in between these nuclear membranes, which are called the nuclear pores that specifically regulate which of the molecules can that specifically regulate nucleocytoplasmic transport, that which molecules from the cytoplasm can come in to the nucleus and which molecules from the nucleus can go out to the cytoplasm. Most importantly, the nucleus definitely harbors the chromosomal DNA, right? Your chromosomes. But your chromosomal DNA exists in the form of chromosomes only during metaphase when they're condensed to a certain extent in a certain manner. Otherwise, they exist in form of this heterochromatin and euchromatin. And one more thing to mention is that the nucleus also has one. Nucleus is membrane bound, but it has one membrane less but assembled compartment, which is called the nucleolus, which also has important functions, especially ribosome biogenesis and rRNA formation. Okay, and nucleus performs so many different functions, right? As we have already learned in our biology classes, that the DNA replication happens within the nucleus, and DNA transcription, that is transcription of the genes by the RNA polymerase to form the transcripts, be it the mRNA transcripts or rRNA, tRNA, and the other RNA transcripts, are done within the nucleus. And also the splicing of these transcripts happens within the nucleus, after which they are exported to these nuclear pores to the cytoplasm, where, for example, the ribosomal machinery is present, which recognizes mRNAs and which translate the mRNAs to the proteins. So this is the nucleus, and there is also considerable cytoskeletal elements, like which is called the nuclear lamine proteins, that form this nuclear lamina that provides mechanical strength to the nuclear membrane. And this is the nuclear lumen, which actually runs continuous to the eventually to the endoplasmic reticulum, right? So next up is mitochondria, right? Which is again very well known and is the powerhouse of the cell. So what do you mean by the powerhouse of the cell is that within mitochondria, two very important cycles to the all the proteins that are necessary, and the very specific setup of biochemistry or exists, which enables you to perform the Krebs cycle and the electron transport chain, which eventually gives you the energy currency of the cell, which is the ATP, right? And I think you all know that within the mitochondria, there are two membranes. It's the outer mitochondrial membrane and the inner mitochondrial membrane. And the inner mitochondrial membrane is quite folded into structures which are called the cristae, which harbor these ATPases, right? Which eventually use the hydrogen, the H plus gradient, the electrochemical H plus gradient to electrochemical H plus gradient that is generated because of the electron transport chain to form the ATP, right? Uh, Basically to polymerize, to phosphorylate, to join one phosphate to ADP to form the ATP, which is the energy currency of the cell. So yeah, and so that's why I think you can appreciate that like this electron transport chain and this Krebs cycle needs so many different unique sets of proteins, mitochondrial proteins that are organized in such a interesting manner in this mitochondrial lumen and this periplasmic space. So in the outer membrane and the inner membrane, and this interesting biochemical setup of establishing this electrochemical H plus gradient because of the electron transport chain, it eventually allows you to uh, basically join ADP and phosphate to form the ATP. And in this electron transport chain, you use oxygen as a terminal electron acceptor, which also holds some significance that we will discuss later in the endosymbiont theory, which is important in terms of the evolution of the mitochondria, how the mitochondria has evolved, right? So in the when we will discuss the endosymbiont theory, we will get to realize that this mitochondria probably once was an independent aerobic bacteria, which the cell engulfed, and then there was a co-evolution between the bacteria and the cell, and this bacteria became a part of, become an organelle of the cell, So basically, this mitochondria also has a genomic DNA. It's called the mitochondrial DNA. Has not should not be called the genomic DNA. Because for the eukaryotic cell, the genomic DNA is basically the chromosomal DNA that is present within the nucleus. The mitochondria also has a DNA on it of its own, has some ribosomes of its own, which are different from the eukaryotic cytoplasmic 8TS ribosomes, and they are more reminiscent of the bacterial ribosomes. Okay, so that is also some. So basically, the eukaryotic cells also have extra chromosomal DNA. I mean, the chromosomal DNA is not the only genome of the eukaryotic cell. The mitochondria has a DNA of its own, as well as has some ribosomes of its own, and some of the mitochondrial genes are actually being expressed by the DNA, by the mitochondrial DNA. Some of the mitochondrial proteins are being expressed by the mitochondrial genes. Uh, that means that genes are present in the mitochondrial DNA, and those many of these proteins are also translated by the mitochondrial ribosomes. Okay, whereas there are also several other mitochondrial proteins which are encoded by the chromosomal DNA that is present in the nucleus. And the proteins are synthesized in the cytosol and eventually delivered to the mitochondria. That is also there. The mitochondria is a very interesting organelle and very important organelle too. And mitochondria's inheritance is also very interesting. Like mitochondria follows a maternal inheritance because during fertilization, it's only the mother's egg which has the mitochondria. The father's sperm doesn't confer mitochondria to the eventually fertilized embryo or the eventually fertilized zygote, which eventually gives rise to the embryo. So it's the mother who gives you the mitochondria. So mitochondria has a maternal inheritance, very uh, precise maternal inheritance. Okay, so now another set of so ribosomes are actually not organelles. Ribosomes are important. They are the translational machineries of the cells. The eukaryotic ribosomes are 80S, consist of the 60S and the 40S, larger and smaller ribosomal subunits. But ribosomes are also not membrane bound. They are membraneless assemblies of proteins and rRNA. So basically ribonucleoprotein assemblies, and they're membraneless. They are usually, sometimes they're referred to as membraneless organelles. But yeah, these ribosomes are ribosomes actually. So ribosomal subunits, these large ribosomal subunits and small ribosomal subunits, they stay freely in the cytosol or in the cytoplasm. Um, Upon binding mRNA and initiation of protein translation, like formation of this nascent polypeptide chain, many a times the ribosomes actually dock onto the endoplasmic reticulum, right? The ribosome will dock onto the endoplasmic reticulum and directly translocate the newly synthesized protein into the endoplasmic reticulum. And because of this docking of ribosomes on the endoplasmic reticulum, so there is a section, this endoplasmic reticulum is called the RAF endoplasmic reticulum. So the endoplasmic reticulum, which is docked by several ribosomes, translating different mRNAs and putting this nascent polypeptide chain directly into the endoplasmic reticulum, makes the endoplasmic reticulum having a kind of rough appearance or these beads, like ribosome appearing as beads on the endoplasmic reticulum that is appearing as a thread type of appearance, and which leads to them, which is referred to as a rough endoplasmic reticulum. However, so as you can already understand from that in this rough endoplasmic reticulum, since these ribosomes, the ribosomes are ribosomes which are bound with these mRNAs, and they are translating the mRNAs, they are synthesizing new proteins, and they are translocating these proteins into the endoplasmic reticulum. So endoplasmic reticulum is the main hub. This rough endoplasmic reticulum is the main hub of protein synthesis, and not only protein synthesis but also protein folding, right? They have a large number of wide or diverse chaperones peptidyl prolyl isomerases, heat shock protein 70s, and different other heat shock proteins that are necessary for protein folding, as well as various different protein post-translational modifications like glycosylation or attachment of some lipid anchors or other post-translational modifications that are necessary to be done for the protein. So for protein, so the graph endoplasmic reticulum are important hubs of protein folding as well as protein post-translational modifications that are eventually necessary for the protein trafficking to the proper cellular compartment as well as proper protein function, right? On the other hand, um, there is also a part of the endoplasmic reticulum which acts as a major hub of lipid and carbohydrate biosynthesis, primarily lipid biosynthesis, and they are not so docked by they are not so docked by the ribosomes. So that section of the endoplasmic reticulum is called the smooth endoplasmic reticulum. Okay. And as I already mentioned, that the endoplasmic reticulum is present in the perinuclear region. That is, they're present just outside the nucleus, just adjacent to the nucleus. And the nuclear membrane is actually a continuation of the endoplasmic reticulum membrane, right? The endoplasmic reticulum eventually gives rise to another stack of membrane-bound flat-like vesicles, which are collectively called the Golgi complex. The Golgi complex the Golgi complex again has three different subsections. One is called the cis Golgi, one is called the medial Golgi, and one is called the trans Golgi. The Golgi which is located more close to the endoplasmic reticulum is called the cis Golgi. And finally, the Golgi which is located more towards the plasma membrane is called the trans Golgi. The Golgi complex acts as a major sorting hub of the cell. That saying that, what I mean to say is that the Golgi complex is like the large railway junctions from which trains come out that move to so many different places, almost connects all the different parts of the country. So here Golgi acts as a major sorting hub from which these vesicles come out that carry the proteins to almost all the different parts of the cells, the mitochondria, plasma membrane, lysosomes, um, and some take cargo even back to the endoplasmic reticulum. So this Golgi plays an important role in ensuring that proper protein cargo is being trafficked to the proper location and in a proper timely manner, so in a proper spatio-temporal manner. Okay, as well as Golgi also, I mean, this protein folding also it plays an important role in protein quality control. Just like the endoplasmic reticulum, the Golgi also plays an important role in protein quality control. So in protein folding as well as protein post-transcriptional modification, like final modifications of the glycosylations, etc. And in terms of protein quality control, both the Golgi and the um, endoplasmic reticulum plays important role that in this fact that if there is unfolding or if there is misfolding of the proteins, right? If there is misfolding of the proteins, then they have to be targeted for degradation by the proteasomal complex or by trafficking them to the lysosomes for degradation. That is also some important functions of the Golgi complex on the endoplasmic reticulum. Lysosomes are those cellular compartments which are quite acidic in nature and are filled with a huge repertoire of hydrolytic enzymes, proteases, lipases, etc., which are necessary for degrading several important macromolecules. It can be protein aggregates, it can be engulfed bacteria which are degraded within the lysosomes, or it can also be cellular proteins which need to be recycled for their turnover. And the small units being small units can be amino acid residues, or in case of lipase, it can be small lipid molecules. So those are again reused by the cell for the cellular physiology. Centrioles and centrosomes are subcellular organelles that become important during cell division for giving rise to the spindle fibers that ensured the segregation of the chromosomes between two daughter cells during the mitotic and meiotic cell division. So they are the nucleation centers for the microtubules, right? They're the nucleation centers for the microtubules, which are integral components of the spindle fibers, um, that as well as they're important for the cytoskeletal organization and reorganization that is necessary for the normal cellular functioning in general as well. Yeah, that leads us to this cellular component. This is not a, I mean, this again, centrioles and cytosols are also membraneless compartments. They are not membrane bound. Okay, lysosomes are membrane bound compartments. Golgi complex is membrane bound compartments. Ribosomes are membraneless, and the centrioles are also membraneless. Centrioles and centrosomes are also membraneless. Cytoskeleton is also an important cellular component, but that is also membraneless, and it's often not referred to as an organelle, but is an important component of the cell. Cytoskeleton consists of, it's like the name truly depicts and describes what its functions are. It's literally, its functions are providing in mechanical strength and rigidity to the cell, to the cellular plasma membrane, and the plays important roles even in the spatial organization of the different cellular compartments like the endoplasmic reticulum, Golgi, mitochondria, nucleus, lysosomes, peroxisomes, etc. Like they become those rods of the cell to which various different things can be anchored, and they also act like these railway tracks on which the vesicles of the cell can move. So they basically act as uh, they they play important roles in the transport and traffic of proteins from one cellular compartment to another cellular compartment. Um, Proteins which are loaded into these vesicles, the cargo proteins which are loaded into the vesicles, which move on those microtubule tracks using various different motor proteins like kinesin, and dynein. So yeah, the cytoskeleton consists of three different compartments: the microtubules, actin filaments, and um, actin filaments, myosin filaments, microfilaments basically. So microtubules, microfilaments, and intermediate filaments. So these three consist of the cytoskeleton. However, as I had already mentioned while discussing about the nucleus, that there are also nuclear cytoskeletal elements, which are nuclear lamine proteins, which form a layer of cytoskeleton lying just inside the nuclear membrane, which provide mechanical strength to the nucleus as well, from the nuclear lamina. Right? And there are also some additional structures. I mean, plant cells have all of these components, except or have all of these components that or subcellular compartments that we had already discussed, but also have some additional compartments like the cell wall, which is made up of cellulose because of the plant cells, which give them much higher mechanical rigidity and also osmotic strength, like provides tolerance against osmotic stress. Even if there is an influx of water, these cells have much more tolerance towards that because of the extra mechanical strength that this cellulose cell wall provides the plant cells. Right? And the lysosomal, the vacuole is kind of the equivalent of the eukaryotic cell of the animal cells lysosomes, right? It's the equivalent of the animal cells lysosomes. But apart from this degradation and recycling functions, the vacuole, because of this plant cell, also plays an important role in water balance within the plant cells because it acts as the water storage organ as well, and also so it plays important roles in maintaining the osmolarity and also acts as a storage organ, like in storage organ in some specialized cells where it stores starch. And most importantly, the plant cells have chloroplasts, which are the hubs for photosynthesis, which is very important. I mean, without which, I mean, all the animals are heterotrophic. They are not capable of photosynthesis. There's the plants who are only capable of photosynthesis, who are the autotrophs, who can harvest the energy from sunlight and can convert them to chemical energy, right? That is food that the animals consume. And actually, um, if we look in terms of evolution, then evolution of these photosynthetic organisms was very important in giving rise, photosynthetic organisms in giving rise to aerobic respiration, which turned out to be eventually very important for the birth of these eukaryotic cells. So eukaryotic cells, since they have such complex organization of cellular biochemistry, so many subcellular compartments, for the regulation and concertedness, cohesiveness, and coordinatedness between so many different compartments, it takes quite higher levels of energy. And so since the eukaryotic cell is so big, it has much higher, it's much bigger as well as much complex. So it requires much higher energy production and energy requirements, has much higher energy requirements as compared to the prokaryotic cells. So, uh, how did, why, or how did the rise of the first photosynthetic cells um, paved the way for the rise of the eukaryotic cells is that because of this rise of the photosynthetic cells, there was evolution. As a result of photosynthesis, there was generation of oxygen into the Earth's atmosphere, which was absent before that. So when oxygen was not there in so high proportion before the advent of photosynthetic organisms, they are the only form of respiration for generating the energy that the organisms needed for survival was the anaerobic respiration, right? So it was only after the advent and evolution of these photosynthetic organisms that there was considerable increase in the fraction or the proportion of oxygen in Earth's atmosphere, which made aerobic respiration possible. And aerobic respiration facilitated the generation of much higher amounts of energy from the same number or same concentration of substrate molecules. And because of this capability of aerobic respiration and generating much higher levels of energy from the same number of substrate molecules, uh, it made possible the evolution of eukaryotes, which had this much higher energy need for organization of for achieving this higher for sustainability and survival of these higher larger eukaryotic cells having higher complexity, having much more complex organization of cellular biochemistry in these membrane bound subcellular organelles, as well as maintaining their coordinatedness. So this brings us to a very important, to very interesting uh, incident, a theory which is called the endosymbiotic theory. So according to that, um, um, that how this mitochondria and the chloroplast, mitochondria of the plant cell and the animal cell both, whereas the chloroplast are present as the plant cells, are very reminiscent of bacterial cells. Okay, so they have two membranes, like in the mitochondria as well as in the chloroplast, they have the outer membrane and the inner membrane, which kind of are reminiscent of those gram-negative bacteria, which have this outer membrane as well as the cell membrane, right? So two membranes in the cell envelope. The ribosomes of the mitochondria and the ribosomes of the mitochondria and the this chloroplast are very reminiscent of the bacterial ribosomes, and there are various other similarities as well. So according to the endosymbiotic theory, what how it goes is like this: that there was one aerobic bacteria, okay, that once the these ancestral eukaryotic cells must have engulfed one aerobic bacteria, that is a bacteria which had the capability of aerobic respiration, right? Before that, the ancestral uh, eukaryotic cell did not have the capability of aerobic respiration. So, but did engulf the bacteria which has the capability, had the capability of aerobic respiration. That is this all these electron transport chain components and maybe also the Krebs cycle components. Um, but instead of degrading this aerobic bacterium, what followed on was a co-evolution of the bacteria and the ancestral eukaryote. And there are also transfer of genes between this bacteria and the eukaryote, the ancestral eukaryotic cell, and eventually this bacteria became like one organelle of the cell, and that which was kept on being passed on from the mother cell to the daughter cell. And next on, so the animal cell, I mean, this animal cell is kind of a result of this animal cell or the heterotrophic eukaryotic cell basically is kind of a result of just this endosymbiotic event. Whereas for the plant cells, this eukaryotic cell may have engulfed one photosynthetic bacteria or cyanobacteria, and that there was a coevolution of that photosynthetic bacteria with this eukaryotic cell, where this photosynthetic bacteria or cyanobacteria has remained as this chloroplast, which has the capability of photosynthesis, harvesting the light energy from sun and converting into the chemical energy because of very important enzymes like Rubisco, using carbon dioxide and the energy from carbon sunlight, which probably gave rise to this photosynthetic eukaryote in terms of plants and algae and others. Okay, so that's what that's the endosymbiotic model of evolution of this modern photosynthetic eukaryote, having both the mitochondria and the chloroplast, and this modern heterotrophic or heterotrophic eukaryote having just the mitochondria as a result of coevolution of the aerobic bacteria. Okay, so that brings us to the end of today's session. If you have any question, then you can ask. Okay. Otherwise, we will meet in the next week, the next session. I hope that you will be
Sir,
able to do your assignments. Shown.
sir, the photosynthetic bacterium you said that will be cyanobacterium or some other photosynthesis type of bacteria.
It can be any photosynthetic bacteria, actually. Cyanobacteria are the major class of photosynthetic bacteria.
Okay, sir.
Hmm. Okay, then. See you in the next session. Bye.
Okay.